Hello everyone, so this is just a walkthrough of your alien genetics activity that we did in virtual school to practice for the final assessment in our genetics unit. So I wanted to walk you through what I was thinking for each of those questions, and we'll start here with looking at the male karyotype. So for this alien species, the males, they have an N of 5. That is to say there's 5 unique chromosomes for this species, and if you look at their diploid somatic cells, their 2N would be 10. So that's what you see here. Chromosomes 1, 2, 3, 4, 5, 6, 7, 8, 9, and 10. Okay, so we're looking at specific genes on specific chromosomes, and I've laid out the different alleles for each individual, in this case, the male and the female. But let's take a look at the male. So if you look at the male on chromosome number 1 here, you'll see that it is heterozygous for the I number gene. So you can see there that one chromosome has a capital dominant E allele, and the other chromosome has the recessive version of that. So with respect to the I number gene on chromosome number 1, this organism has a heterozygous genotype. Okay, so moving right along, this is the rest of this male's genotype. Okay, so when I look at this, I can convert this genotype into phenotypes using this allele tracker over here. So because it's heterozygous, it's going to have 2 I's. Because it's homozygous recessive for the horn number gene, it'll have no horns. Keep in mind, I'm just taking what I see over here as a genotype and going to the specific gene for it and looking at what the alleles create phenotypically. Okay, so it'll have no horns because it's heterozygous for the smell gene. It'll be smelly. Again, that's because if you have one dominant S, which this one does, it'll have that phenotype of smell. For the skin pattern gene, it's homozygous dominant, so it's going to be striped. And then with respect to the last gene, the one that's on chromosome number 5 here, we'll use this gene tracker over here, and it's a heterozygote, so it has the non-circled capital C paired with a lowercase that is the underlined C. So in this case, the phenotype would be blue skin. Okay, so this male has 2 eyes, no horns, it smells, has striped skin that is blue. Okay. Moving along to the female, so now we'll look over here at this part of the slide, and we'll write down the female's genotypes. So she is the following. Okay, so what does that break down into phenotypically? Because she's homozygous recessive for the I number gene, she has 3 eyes. Because she's homozygous dominant for horns, she has 2 horns. Because she's heterozygous for the smell, she's smelly. Because she's heterozygous for the skin pattern gene, she's striped. And now she has an interesting phenomenon on her chromosome 2, chromosome number 5s, because she has these 2 co-dominant alleles, one that would create blue and one that would create yellow. So a little tricky there when you mix blue and yellow. Her skin phenotype would be green, because you see both of those alleles creating their phenotypes together. Okay, so it blend together. Okay, so moving right along, the alien species here, I just have a few questions about it, and I sort of gave it away in the previous slide, but if you're looking at these, you see a pair of homologous chromosome number 1s, pair of 2s, 3s, 4s, and 5s. So the N equals 5. So that is to say, when that male creates sperm, there'll be 5 chromosomes, and a number 1, a number 2, a number 3, a number 4, and a number 5 in that gamete. But if you look at a diploid cell like the one that we're looking at now that has pairs of homologous chromosomes, you'd see 10 chromosomes. Okay, so when you go on to play with this, just make sure that you're understanding the difference between haploid and diploid, N and 2N. So now we're going to do a few crosses. So one of the first crosses that I ask you to do is just to look at the I gene. Okay, so that I number gene, again shown right here, is going to ask you about a cross. So we're going to cross this male to this female. And so, you know, by convention, we've been putting this into Punnett squares. So what this means is this male can create sperm that have on their chromosome number 1 either a capital E dominant allele. The other half can have a recessive allele for that I number gene on chromosome number 1. So it's a 50-50 whether or not you get capital E or lowercase e from this male as an offspring. The female over here, she's homozygous recessive, so the only thing that she can put into her eggs is that lowercase recessive allele. So you remember, you don't have to draw a giant Punnett square to do this. It's a 2-box Punnett square, and half of their kids will be heterozygous and half will be homozygous recessive. So it's a 50-50 if you look like mom or dad in this. So that is to say 50% phenotypically would have 2 eyes, whereas 50% phenotypically, remember I'm using this information right here, would have 3 eyes. Okay, moving right along. The next cross we're going to look at is for the horn gene. Our horn gene is over here on chromosome number 2. And we just basically look at the phenotype, or the genotypes. So dad is homozygous recessive, mom is homozygous dominant. It's the easiest Punnett square to set up in the world because the only thing dad can package into a sperm are chromosome number 2s that have the little h allele for the horn number gene. Okay, so you get little h there. Mom is only bringing big H, so 100% of their offspring would be heterozygous. That is to say, phenotypically, 100% would have 2 horns. Okay, so nobody would look like dad in that case. Smell gene. Let's do a cross for the smell gene. So in this case, smell gene is on chromosome number 3, and the capital S creates smell, whereas the lowercase recessive s does not create smell. So in this case, for this cross of these two individuals, both individuals are heterozygous. So dad will have a 50-50 split of what he gives his offspring. Mom will have a 50-50 split. And this is one of your more conventional Punnett squares. So 75%, that is to say this one, and this one will have at least one dominant capital S allele. So 75% would be smelly, like their parents, and that lucky, I guess, 25% shown right here would not have a smell. Moving on to the skin pattern gene. So again, just following the genotypes. Here you'd have a male that gives 100% capital, that is to say dominant P allele to their offspring, whereas the mother in this case, it's 50-50 what she gives her offspring because she's heterozygous here. So that is to say 50% will be homozygous dominant, 50% will be heterozygous. But if you look at the gene tracker over here, if you have one capital P dominant